Hi everyone. Today I'm going to go into concepts related to the most common question I hear all the time in clinical practice working with chronic patients, which is, why do we get food sensitivities? Why are the rates of food sensitivities going up? So I want to try to explain it to you from an immunological perspective. So what I want to do is the first 15-20 minutes, I want to give you some background information on those concepts, and then we can open up a question and answer, and I'll be happy to answer any questions anyone has related to the topic. So I think we've all seen that we've had a significant number of food sensitivities develop in different populations. I think for most of us that or adults, when we were kids, we didn't see all these kids sensitive to nuts and having different sections in school lunch areas where kids were not permitted to bring any types of nuts or especially peanuts. And we've seen an explosion of autoimmune disease, and current research shows about 20% of the worldwide population has confirmed that they know about some adverse reaction to foods. So the question is, why are these things happening? And what I want to do is I want to try to explain it to you from the peer-reviewed immunology literature. And then I'll also share with you some concepts and theories that are out there that haven't been proven yet, but just some of the theoretical models that need to be investigated in the future. And these models I'm going to share with you are from the immunological world, meaning immunologists that are working on food protein sensitivity or involved with it. So let's first talk about the difference between food sensitivity versus allergy. That's the first place to start. So food allergy is different than food sensitivity. And the reason that's different is because of the type of immune response. So when we have an immune response to a food protein, there's certain antibodies that are formed to cause that immune reaction. And one of the antibody that's used in an allergy is called an IgE antibody. And the IgE antibody is fast, it's immediate, it activates what are called mast cells, which get high amounts of histamine, and that's your classic allergy. Whether someone is allergic to dust or dander or certain things in the air like pollen that have immediate reactions, those are IgE sensitivity. And those are the ones that classical allergists are concerned about, because they're not really concerned about food sensitivities or sensitivities, are worried about allergy. And the reason why is also because allergies can cause anaphylaxis and cause the throat to close up and cause some serious inflammatory reactions. So in the conventional world of allergy, the focus is really on this IgE, so-called immediate immune response, because it's such an aggressive response. And those types of food sensitivities can be measured with blood tests, where the antibody that's measured is IgE, or they can also do what's called a skin test, where they prick a person with different proteins and see if there's a skin reaction, which is a very unreliable test, which I don't understand why people are still using because of the data out on it. Now, then you have what's called food sensitivity, and food sensitivity doesn't involve this IgE antibody reaction. So these the other antibodies involved with the food sensitivity are IgG, IgM, and IgA, and these are called delayed reactions. And the difference between them is that the response isn't as aggressive, is not as fast, but it does create this inflammatory cascade. And these types of food sensitivities, not allergies, are measured with blood tests, where you look at a food protein and they're measuring one of these delayed antibody markers. So it could be, for example, corn IgG, or corn IgM, or corn IgA. So those are the key difference between food allergy and food sensitivities. And you have to understand that tests that measure IgG, IgM, or IgA are not testing allergy, and they're only testing food sensitivity. Now, food sensitivities, when they get active enough and aggressive enough, they can not cause the delayed reaction. They can be pretty immediate. So there are people that have food sensitivities to, for example, milk protein. As soon as they eat it, they get significant inflammation and bloating, and they don't have an IgE allergy reaction, but they have a food sensitivity that is now so aggressive that it almost looks like a immediate onset, that they have symptoms within the first few minutes. Now, the problem with food sensitivities are sometimes when you get exposed, the inflammatory responses are very subtle, and then they create inflammation over the next few days. And then if you have multiple food sensitivities, it's really hard to identify them. This is why sometimes people just go on a extreme diet elimination diet where they get rid of, let's say, an autoimmune paleo diet where they get rid of all foods and then they see if they feel better. There's certain food proteins that are more reactive than other food proteins. They get rid of those, which are typically milk, wheat, grains, nightshades, and then they see if they feel better. So that's just the basic background. So just to make sure we understand the terminology. Now let's talk about why do people actually have more food sensitivities. Well, the reason why people have more sensitivities is because of two main reasons, and this is just from classical immunology. The antigen has greater antigenicity, or there's increased immune reactivity by the immune system. So let me explain each of those concepts in more detail. So the antigen is referred to something that's foreign. So anything that's foreign to the human body is that enters and has the immune system react against it is called an antigen. And the antigen can be a pathogen, like a bacteria or a virus, but food proteins are also called antigens. Now, there's a concept of antigenicity, which is how aggressive that antigen is and how reactive that antigen is. And we know right now the most common food proteins that have the highest degree of antigenicity or the highest degree of reactivity in people tend to be gluten, milk proteins, albumin, which is egg protein, casein from milk, and we're also seeing lectin proteins become very reactive, to, especially in autoimmune disease populations. So those food proteins typically are the ones that, in a clinical setting, when we're suspecting a person may be having an inflammatory response from their diet, those are the ones to suspect first. Now, you can have a food sensitivity literally to any protein. But the key concept is the antigen itself. Now, one of the things you have to understand about antigens and the immune response against foods is this antigen that comes in as a protein. You can't develop a food sensitivity to vitamins or minerals or isolated sugars or fats. It has to have a protein with it. Now, it could be a fat with protein like a lipoprotein or a sugar with protein like a glycoprotein, but for the most part, antigens have to have a protein structure where the immune response reacts against the protein and it makes antibodies. And the immune system doesn't react to the entire food protein or the entire protein. It only reacts to a branch of the protein. So think of a dietary protein as a tree with many, many branches. And each branch is what they call an epitope. And each branch of these epitopes have amino acids. And then there's an amino acid sequence for each of these branches of the protein. So what happens in immune responses to food proteins is the immune system decides to react against one of those branches, one of those amino acid sequences, not to the entire protein. This is why, for example, if people have gluten sensitivity, if they only measure alpha-gliadin, they may not catch the gluten sensitivity. They may need to check gamma-gliadin or deamidate-gliadin or gliadin-17 mer. There's different branches of the protein. Or if someone reacts to milk, they may need to not just measure casein, but they may have to measure butyrophilin, or they may have to measure caseomorphin. These are different protein portions that are involved with milk. So each food protein has different branches, and when we do a sensitivity test in the lab, they're looking to see if there's a antibody elevation to that food protein. But as far as you see, when you look at the results, all you see is like corn or soy or egg or grape or whatever, pineapple antibody, either IgE, IgG, IgM, or IgA, and that'll give you a clue what's going on. So this concept of antigenicity is basically based on there's a protein, we respond to some part of that protein. Now, the concept that I was trying to explain earlier that I want to get into is that what determines the antigenicity, how aggressive the immune protein is, can be changed by a couple things. So one of the key things is if the antigen changes its configuration. If the configuration of the protein changes, it can become either more reactive or less reactive. And research studies have found that the protein configuration can change when heated or when cooked. So some foods become more reactive or less reactive or more antigenic or less antigenic when heat's involved. So some people may react to raw eggs on a food allergy panel, but they actually only, but their response is actually to cooked eggs. Or someone can react to raw tuna versus cooked tuna. So we know that heating can change the configuration. And this is very important to understand, because when you're looking at antigenicity, you have to realize that when you're looking at conventional laboratories, when they're measuring foods, they're all raw. They're all measuring raw food. So lots of food sensitivities are missed with classical food sensitivity testing, because all of the labs are using standardized kits where they're using raw food proteins. The only lab in the world that's doing raw and cooked, because of the research and because they're the leaders, is a lab called SIREX. And that's really the only way you can find the difference between antigenicity. So one of the key things to understand is if you have a reaction to food, it may be different if it's cooked versus raw. The other thing that's interesting is the research has found, in a great article published by immunologist Dr. Istafajidani, he found that when foods combine together, they actually also change the antigenicity, or the immune reactivity, of the food protein exposure. So, for example, he tested all the different ingredients in a pepperoni pizza individually, and then just tested pepperoni pizza, and then saw that some people there's a significant reaction to people that just reacted to pepperoni pizza instead of each of the ingredients. He did the same thing with donuts, he did the same thing with several other food proteins, but he showed the individual ingredients may have some degree of reactivity, but when they're all combined together, they may have more, because when these proteins are all cooked and heated together, their structure changes differently. And what they do, by the way, in the lab is they take a food protein, they grind it up, they purify it, they put it on a well called a ELISA well, and then they take the patient's blood, they mix it with the well, and then they put a dye into that well that sees if there's any binding, and then they measure the degree of color change with the dye with what's called an optical density measurement machine, and then they can see how significant the reaction was, or if any reaction occurred at all. So that's called ELISA testing. So he did that with individual foods and with combined foods, and saw that there was a difference. So one of the interesting things is that when you look at the concept of food sensitivity, that the combining of food proteins together may change the antigenicity, and cooked versus raw may change the antigenicity. And then ultimately the question becomes, okay, so if we're seeing an increased raise in food sensitivities, what's causing these food proteins to be more antigenic? Why are they being so much more reactive than now? So there's several reasons for this. So let's talk about, for example, wheat protein. There's been an explosion of gluten sensitivity in the US and Europe, and that's noted. I'm sure it's worldwide. And what they have done clearly with some research to show that it's not really the increase in the prevalence of gluten sensitivity is not because it's a trendy diagnosis. They've actually looked at blood samples of soldiers in War and Air Force Base from 50 years ago to now, and they did a gender and age-matched comparison, and they found a significant increase in the rates of gluten sensitivity to populations 50 years ago to now. So in the world of immunology, in the world of celiac disease, they've actually investigated this question, and one of the things that they've noticed is that the gluten compounds, the gluten proteins, for example, which is an example of one food protein that's more antigenic now than before, has been hybridized. That, one of the things that farmers have done is they've changed their seeds. They've mixed seeds together to the point where they can be more weather resistant. And some studies have shown, where they do spectroscopy, that there's a 3 to 5% difference in protein sequencing with what's called native wheat versus modern wheat. And these are terms that are used in the immunology literature: native wheat versus modern wheat. So modern wheat is tend to be more antigenic, more immune reactive. So we know just the fact that they're not genetically modified, but they've been hybridized, that these food proteins are much more different now than they were before, specifically with wheat. The other key thing that some immunology researchers have theorized, and most of this work has come from MIT researcher Stephanie Seneff, Seneff. She's really, put together some great research and some great information with the role of how pesticides, specifically glyphosates, bind to wheat. And when a chemical binds to a protein, it changes the configuration of the protein, and it becomes more antigenic. That's actually what my PhD was about in immunology. I did it with fire retardants and serum albumin, and found when people get exposed to fire retardants bind to the protein in blood called serum albumin. They change the configuration, makes it more immune reactivity. Well, so there's some theories and some studies that have shown the spikes of reactions with celiac disease and gluten sensitivity to the usage of different pesticides, specifically glyphosates, according to the Professor Stephanie Seneff's work at MIT. So we know that that's one possibility. And the use of pesticides and how they use pesticides to clear fields have dramatically changed over the last few decades. So one of the theories in the world of immunology why there's so much more food sensitivity is that these pesticides are not just washed off. These pesticides bind to proteins. A great example of a food protein binding to a chemical binding to a protein is if you ever use a lollipop and lick your tongue and your tongue turns red. That's food coloring binding to protein. That's a chemical binding to protein, it just stays there. So when pesticides bind to different types of grains, they change the configuration of protein. They're not simply just washed off. And then those proteins with a new configuration have the potential to cause some degree of immune reactivity. So that is one of the theories of why some foods are more antigenic. Also, there's research that shows, and I'm going to stick with gluten because a lot of the significant research has been done with gluten, but it could be with any food protein. The concepts are the same. So in addition to things like chemicals or pesticides binding to proteins, changing configuration, making them more antigenic, the other key thing is that proteins, when they go through a food processing mechanism where they're adding different chemicals to make it more flexible or more stable, or adding different types of gums to process the food. For example, someone taking a basic grain and then adding different things to make it a pretzel that make it more sticky or less sticky or more flexible, or change the structure of it. Those things bind to the proteins and change the structure of it, and they can become more reactive. So, for example, deamination takes place with gluten. When gluten is processed into things like pretzels or cookies or other compounds, the food processing of it changes the configuration of the protein to not just what's alpha gliadin, but what's called deaminated gliadin, and now that's more reactive than before. So that's another major issue with foods, that the chemical process of food processing is changing the configuration of the protein, making it more antigenic. And then a third mechanism that's out there related to food sensitivities is related to food coloring. And food coloring, and one of the things interesting about food coloring is that food color itself is not going to cause an antibody response, but food coloring binds to a protein. And when it binds to a protein, it prevents that protein from being broken down. And remember, when your proteins or different branches, if they stay in that structure and they make antibodies against them, then the inflammatory immune response takes place. But a food protein has to eventually be broken down by digestive enzymes into individual amino acids, and when they're broken down to individual amino acids, instead of an entire combination of amino acids, which is a protein, then they lose their reactivity. So as long as the food protein gets undigested, it's going to constantly cause reactions. Well, food coloring significantly impacts the breakdown of food proteins. So, I mean, the worst scenario is when you look at antigenicity, is you take a food protein that's been hybridized or even genetically modified that can change the protein structure of it and make it an issue for susceptible people, combination with food processing, or that also changes the configuration of the protein, and then add food coloring to it. And now you have a food protein that's very new, it's a new antigen, may have greater antigenicity and be hard for the system to break down, and now you've got a major trigger, so you have a greater degree of antigenicity. So just to give you the big picture, because I have a few more things to talk about before I take questions, is that food proteins, the reason why we're seeing so much elevation in food proteins is because of two reasons. One is the antigenicity concept, which is chemical pesticides binding to the chemicals, food processing, food coloring. Then the second issue is how the immune system reacts to food proteins. Okay, so let's talk about how increased immune reactivity to a food protein is also part of the reason why we're seeing a greater degree of food sensitivities. So when we ingest a food protein, the minute we ingest it, we have immune cells in our mouth that are sampling the food proteins that are called dendritic cells. And these dendritic cells immediately tell the immune system, hey, there's a new food protein coming in there. There is either a response or not a response against it. And then once we swallow the food protein, those food proteins get into our gut, as a solid protein, and then digestive enzymes have to break it down so that it becomes much reactive. But we also have food protein sampling it, and then eventually our food proteins have to be broken down to very, very, very small amino acids so they can cross the intestinal lining, and then they get into our circulation and bloodstream. Then we have T cells and other immune cells in our bloodstream that are sampling and reacting to the proteins. And if any part of this cascade is dysfunctional, then you can have increased reactivity to proteins. So the most popular one that's out there as a mechanism that causes a person to be themselves more reactive to the food is just the concept of leaky gut or intestinal permeability. That's where the intestinal tight junctions open up, and when intestinal tight junctions open up, then undigested food proteins can cross, and as soon as those undigested food proteins cross, then immune cells react against it. That's the major concept of leaky gut. And when people have leaky gut, where their intestinal tight membrane junctions open up, then they can start to have the development of many, many new foods, unrelated to the degree of antigenicity. It's its own independent factor. So there's been an explosion of research with intestinal permeability in the past 10 years, and the thing is many people develop intestinal permeability when they look at studies because they have lots of intestinal inflammation. We for sure know celiac disease patients have intestinal permeability. We know autoimmune disease patients have intestinal permeability. And these inflammatory responses open up. We recently published a paper in the Journal of Molecular Science where we looked at patients with inflammatory bowel reactions. We found a really high percentage of them had intestinal permeability and blood-brain barrier permeability. So we know that in inflammatory conditions, when there's gut inflammation, when there's potentially bad diets, when there's lots of stress, those things all impact the integrity of the tight junctions, and when they open up, that can cause food sensitivity. Another key thing that's causing people to react more to foods unrelated to leaky gut is their microbiome. And we know that when a food protein eventually gets into the gastrointestinal tract, that different bacteria in the gut will either dictate a response against that food protein or not, or modulate the effect of T cells or other immune cells in the gut and dendritic cells to either respond or not respond against it. And bacteria in the gut produce something called polysaccharides or postbiotics, which signal the immune system. So when people have really poor microbiome diversity because they're eating the same food every day, or they're eating fast food every day, and they're not getting enough healthy, diverse vegetables into their system, their gut microbiome can change, and then they can be more reactive to foods and unable to calm down the inflammatory response when they're eating foods that are inflammatory. Another main mechanism that can cause a person to be overreactive to food proteins, which is why they're developing food sensitivities, is something called their SIGA levels drop. Their SIGA levels drop is a key factor. So let me explain what SIGA is. In your gut, you have these immune cells called secretory IgA cells. And when you eat food proteins, these SIGA cells engulf the food protein, and when they engulf the food protein, they basically prevent it from constantly activating immune cells. So it's like a layer of coating to calm down the immune response. And studies have shown that significant stress, overtraining. So in one study, they looked at athletes going through basic training. Soldiers going through basic training. Had their SIGA levels significantly drop. So those are some reasons why you can have increased responses to food sensitivities. And then what they've also found is that there's an intimate connection between immune cells in the microbiome and gut and immune cells in the liver through a pathway called enterohepatic circulation, in which if these macrophages or immune cells in the liver are overactivated with constant, for example, environmental exposure or chemicals, that can make the gut immune systems more responsive. So those are some of the main mechanisms that cause these immune reactivities. Now, what happens to a lot of people is something takes place. They get an autoimmune disease that develops all of a sudden. And when they get an autoimmune disease, they start to react against their immune tolerance gets compromised. And this is another concept. Everything I talked to you about intestinal permeability and microbiome imbalances and overactive dendritic cells and SIGA, overactive Kupffer cells, are related to a concept called immune tolerance. And by the way, if you go to Dr. K News, I've written several articles about immune tolerance and these concepts. And I do have an online program that teaches you how to improve your immune tolerance. It's called the 3D Immune Tolerance Program. So if you're interested in learning more, please check that out. But let's go back to the basic concepts. Now, at some point, someone may get an infection, and then their immune system gets dysregulated, and whether their SIG levels go down, or their dendritic cells become dysfunctional, or they get intestinal permeability, now they have multiple food sensitivities. Someone else can get severe stress, and their immune system dysfunctions. Someone else can be exposed to a chemical load, and their antioxidants get depleted, and their gut barriers get compromised. Their Kupffer cells in the liver get overactive. Now they develop food sensitivities. And sometimes the combination of all these factors. Someone could be living in a mold-infested building for a period of time, and over the next year or two, they get that constant mycotoxin exposure that breaks down their barriers and causes problems. We have created a live chat on that as well. So those are all the basic concepts that are involved. Now, one last thing before I take questions, which is really important. I'm sorry I'm going a little bit over than I wanted to, but these are things it's hard to just have a discussion unless you understand these fundamentals. There's also a concept called cross-reactivity. And cross-reactivity means one food protein or antigen has a similar amino acid sequence to another. So when a person makes antibodies against this one food protein, it can also cause reactions against things that have the similar amino acid antibodies. And they first identified cross-reactivity in the immunological literature in 1942, and what they found was that certain people that were sensitive to pollen allergens had sensitivities to specific fruits. And then what they found was that the amino acid sequences of certain fruits were identical to pollen, and then they had this cross-reactivity response. And this takes place with, for example, nuts. Most nuts have similar amino acid sequences, so when you get exposed sensitive to one nut, you get sensitive to most of them. Same with seafood. Most seafoods have similar amino acid sequences, so when you get exposed to seafood proteins like crab or lobster or salmon, you start to react against other food proteins. That's why many people say they have a nut allergy or they have a seafood allergy. It's because the amino acid sequences of these food proteins are so similar that once you develop sensitivity to one of them, you react against all the other ones. So those are all the different key concepts with intestinal permeability, food sensitivity. And what I'll try to do is I'll try to get into some questions here and go from there. Okay. The only thing is when I try to look at these questions, I won't be able to answer questions that are case studies. I can't manage a case through these. Yes, so the question from Heather is, why will foods come up as reactive on testing if the person has not eaten it? Random foods such as lobster, where you know there isn't any cross-contamination with other foods. Well, Heather, this is a good question. So, when you look at food antibodies and food proteins and food allergy testing, if you get exposed to a food protein, antibodies are going to spike up in the first 2 weeks, and after about 2 weeks you get a good reading of them. For food sensitivities, IgG, IgM, IgA, and then they'll stay up for a few months, maybe about 2 months, 2 to 4 months. So if you haven't been eating the food protein, then there's no reason for it to show up on a lab test. But if you've been, for example, if you tested positive for lobster but hate lobster and never eat lobster, and you're wondering why the food sensitivity test is showing you that you have reactions to lobster, even though you haven't eaten it and never have eaten it because you hate it your whole life, or I don't know, in the past 10 years, you may be eating shrimp, you may be eating another seafood or another food protein that's similar in structure, so it's going to show up on your lab test as if you were eating that food protein. So the reason why you may have foods show up that you're not eating is because of the cross-reactivity with sensitivities to foods you have that have similar structure proteins. Okay. Let's go into next question here. By the way, thank you for everyone for joining me. It's always so nice to see people interested in these topics. Okay. Is there a type of protein powder you should stay away from, and are there any types that can be recommended from Asuka? Well, I can tell you for the most part, most food proteins have an issue. So when you look at whey protein or you look at protein powder drink, whether it's pea protein or vegetable protein or collagen protein, whatever, there's a possibility that they may be an issue because the processing of a food into a powder changes the configuration of the protein, makes it more antigenic. So I know for myself, when I work with lots of autoimmune disease patients, I don't recommend any powders for them because most of them will react to powders. And if you are a person that has lots of food sensitivities and your immune tolerance is weak, you really shouldn't be eating any foods with protein powders, protein powder shakes, because they have a tendency to be very antigenic. So be aware of that. It's just better to eat whole food. Once you start to develop lots of food sensitivities, you really need to stay away from high antigenic foods. Any protein that's been processed, like protein powders, is something you want to be very careful with. Just like you also want to be careful with eating proteins that have lots of food dye on them, because if you can't break down those food proteins because of the food dye, that'll increase the immune response against it. Okay, some questions here. My wife's Dr. Reyes is here to help me.
Hi,
Thank
Dr.
you.
Carlson. All right, here we go. From Susan. I understand your 3D immune tolerance program helps those with low SIGA levels. I've had high SIGA levels for a year.
Yes.
Can the 3D immune tolerance help with that?
So the question is, we talk about low SIGA being a factor with causing increased immune sensitivity. What about high SIGA? Well, the 3D immune tolerance program, by the way, is the course that we teach. The 3Ds stand for diversify, distinguish, and downregulate, which are concepts we talk about to implement to try to improve immune tolerance. But if you have high SIGA, then you probably are dealing with an active trigger, and you probably have either an infection in the gut that's still not identified, or you're still getting a significant reaction to food protein. It could be you could be gluten sensitive and still get trace amounts of gluten exposure. SIGA is telling you that there is a active trigger in your gut. So the most important thing is to try to identify that trigger and remove it. You can still implement all the concepts of the 3D immune tolerance. It'll just help make your immune system and your microbiome healthier. But at the end of the day, if your SIGA levels are high, then you have an active inflammatory trigger that you still have not identified that you need to remove.
Okay. So MJ, this is something you said previously. Are you saying a food sensitivity reaction can't be fast or cause anaphylaxis?
So a food sensitivity reaction cannot cause anaphylaxis. That has to be an IgE, but immune sensitivity reaction can be immediate over a period of time. If the memory B cells, start to become very efficient to that dietary protein, then as soon as you get exposed to the dietary food protein and have a sensitivity reaction, meaning with IgG, IgM, or IgA, then you can have an immediate food response. So you can have that happen. Typically, when you look at the classical model of food sensitivity, they call it a delayed reaction because from a lot of these food proteins, it takes a day or two to have that immune cascade take place. But over time, it can definitely become almost instantaneous.
Okay. So David's asking, how can you get enough calcium in your diet?
How can you get enough calcium in your diet?
Mm-hmm.
Eat vegetables. Vegetables are really, really high in calcium. Please don't think you need to drink milk to get calcium. Just be aware of that. I mean, you don't need to drink milk to get calcium. You get plenty of calcium if you eat a lot of healthy vegetables.
Okay. Deborah, you kind of answered this before, but you might want to again. Okay, if you haven't eaten gluten for years, can you get an accurate test showing you how to have a sensitivity to it, or do you need to eat it before the test?
If you haven't eaten gluten for many years, there's a strong possibility you may get what's called a false negative test. So you haven't had the exposure to the food protein, so you're not going to have antibodies against it. Typically what we find with people, though, even if they've been on a gluten-free diet for a period of time, they're still eating foods that are cross-reactive to wheat accidentally, like other grains. Other grains have been found to cross-react to gluten, and milk protein as well. And if you get exposed to those foods, since they're so similar to gluten, you can still see gluten antibodies show up. Now, for those people that have been completely gluten-free, grain-free, dairy-free, it's a very strong possibility if you've been very diligent in reducing your exposure to gluten, you may get a false negative test.
Okay. Gina, is there a difference between IgG food sensitivity and IgG4?
Is there a difference between food sensitivity testing with IgG and IG?
Is there a difference between IgG food sensitivity and IgG4?
Immunoglobulin G is the antibody that's being checked. There's different ways to measure it. I don't know the difference in sensitivity or specificity or reproducibility between one or the other. So that's the best I can answer it. But ultimately, it's a delayed food sensitivity reaction.
Okay. Patrick, should you be eating the foods you want? Oh, wait, sorry, that's already. Okay. Okay, Jonathan. People say SIBO or Candida can be the cause of food sensitivity. Which one is the most likely culprit, SIBO or Candida, if you believe that?
Okay, well, for sure, SIBO can absolutely cause food sensitivities because it completely impacts the immune system and the immune tolerance. When you get a bacterial overgrowth in the small intestine, those bacterial overgrowths that then release gases also create an inflammatory environment in the gut that opens up tight junctions, and you can definitely upregulate the immune system. So without question, people that have small intestinal bacterial overgrowth that have reactions to food that are not just antibody based, they set the structure of losing their tolerance, where they also then get, in addition to not being able to digest sugars and starches from the bacteria that's in their small intestine, into causing things like intestinal permeability, loss of tolerance, where they have antibody reactions to foods. Candida is secondary to something else. The concept of having Candida, you should always ask, Candida is secondary to what? Candida itself is not going to cause any kind of food sensitivity by itself. Now, there's some foods that can cross-react with Candida antibodies. So if you're reacting against Candida because you have an overgrowth and you have high levels of Candida antibodies, you may be more reactive to yeast proteins because of cross-reactivity. So there's some degree of cross-reactivity, but for the most part, you always want to investigate what's the cause of the Candida in that scenario. Okay, I think I answered that.
Okay. Suma, is there an old-fashioned way to test for food sensitivities without going to the lab?
Is there an old-fashioned? Okay, there is an old-fashioned way to test for sensitivities. It's actually the gold standard. So the gold standard, meaning that the most effective way to look for food sensitivities, isn't actually a lab test. It's actually elimination provocation. So you eliminate the food protein for 3 weeks, and then you add it back in. So this is a really good way, if you don't have access to lab tests and you are suspicious about certain foods, of what kind of diet you should have. So the easiest way to learn about this is just go online and follow the guidelines of something like the autoimmune paleo diet. And the autoimmune paleo diet really does a great job of removing foods that are very reactive. And I have articles on this on my website, Dr. K News. But when you look at the autoimmune paleo diet, you're removing gluten, dairy, all grains, and you're removing what are called nightshades. And nightshades are foods that have very high lectins, and you're also removing lectins, foods that have seeds like tomato, eggplant, potato. So when you go off those foods for 3 weeks, the first thing you want to determine is, do you notice your symptoms go away? Do you notice your swelling go away? Do you notice inflammation go away? Do you notice your joint pain go away? Do you notice your brain fog go away? Do you notice your energy improve? And if the answer is yes, after, again, you can't do it within 2 days, after 3 weeks, then there's something that you're reacting to. And then what you want to do is add in one food protein every 3 days to see if you get a reaction. The first few food proteins you add in first will give you the biggest indication of whether you react to it. And part of the reason is because when you've gone off these reactive foods that you have withdrawn from your diet for 3 weeks, your mast cells and your immune cells are just swollen and ready to go. They haven't been used, and you can have actually a more severe reaction than you've ever noticed to a food protein after an elimination diet. Don't misinterpret that the elimination diet has caused you to now severely react to the food. It's just because it's the first one you put in after taking a big break from your immune system. Over time, it's going to go back down to the other one. But the first 2 or 3 foods you add in will be the most sensitive foods. So those are strategically really think, what do you want to add in first that you're not suspicious about? As you keep adding your food proteins in, you kind of lose your window. And maybe after adding 5 or 6 foods back in, you may be all swollen and inflamed, and then you can't add in foods again to determine if they're reacting with you anymore, because you're already so swollen and inflamed. And you may have to go through another 3-week period of time to see if that is an issue with you. So elimination provocation diet, it really is one of the best ways to determine if you react to foods. And remember the concept of cooked versus raw and how foods combine together. You could specifically react, for example, to one form of tandoori chicken by one restaurant and not respond to another one because of the spices and foods they use and the food coloring they may use. So be aware of those concepts too.
Okay, Annika, if you clear SIBO, can you tolerate FODMAPs again, or will you always be sensitive? And can you do a SIBO Facebook Live? Haven't you done a SIBO Facebook Live?
No, I've not done a SIBO Facebook Live, but thank you. That might be a good topic. So small intestinal bacterial overgrowth is a complicated condition, and it really involves dysfunction in the gastrointestinal nervous system where the valves aren't working and there's impaired motility for the majority of cases. I mean, you can get it from hypochlorhydria, you can get it from surgery, from changing anatomy, but for the most part, the gut nervous system, which should control valves, isn't working. So bacteria from the large intestine move into the small intestine because these valves aren't working. And then you get these bacteria that are very reactive to sugars and starches, and this is why people go on what's called a FODMAP diet, which is a diet where you avoid fructose and oligosaccharides and disaccharides and monosaccharides and polysaccharides. It has the word FODMAP. You avoid all those basic sugary foods that feed that bacteria that is in the small intestine now, and that causes bloating, distension, and inflammation. If you can figure out the cause of SIBO and you eliminate it, then fine. Then you don't have to be on a FODMAP diet. If the bacteria from the larger intestine stop moving into the small intestine, you don't need to be on a FODMAP diet. But most patients cannot resolve it. And the recurrence rate, even with conventional therapy where they use antibiotics and what are called prokinetic drugs to improve intestinal motility, is very poor. It's almost like 98% recurrence. So it's pretty complicated. Actually, we probably should do it as a separate talk, but the concept is if you don't have bacteria going from your normal healthy bacteria from the large intestine translocating into your small intestine, you would have no further need to be on a FODMAP diet.
Okay. Monique, if you tested negative for celiac but are found to have gluten sensitivity, can you still experience symptoms of celiac, such as unexplained weight loss,
yes,
etc.? So
the question is, if you have tested negative for celiac disease but positive for gluten, but you have confirmed gluten sensitivity, can you have as severe reactions?
Can you still have symptoms?
Can you still have symptoms?
Celiac,
yes.
like weight loss
yes.
and unexplained that kind of
yes.
stuff.
So yes, the answer is, so gluten sensitivity itself is much more severe than people have thought. And they used to clearly, I don't know, this is maybe 15, 20 years ago, the gastroenterology literature would be like, if it's not celiac disease, gluten cannot do anything. It's not a big deal. You have to have the genotype, you have to have this HLA-DQ2 and 8, you have to have positive biopsy, and if you have all those things combined together, you have celiac. That's the only time gluten can be an issue. And that was maybe what they thought 20 years ago. But now what they found is that's not accurate. And gluten sensitivity itself is a significant, very inflammatory condition. And what they've actually found is that gluten sensitivity, not celiac disease, but gluten sensitivity causes significant brain inflammation in large subsets of populations, and several studies have shown this. So if you're sensitive to gluten, you can have significant inflammation throughout your body and symptoms like weight loss and headaches and chronic inflammation and basically the gamut of things that are caused by chronic inflammation. So it doesn't have to be celiac. Now, if you do have actual celiac disease, you have a significantly different immune response to gluten. And what happens in actual genetic genotypes of celiac disease is that there is a very specific exaggerated response with T cells that cause significant inflammation and destruction of the intestinal lining and increase the risk for autoimmune disease dramatically. So celiac disease is much worse because it has an overactive T cell response, but gluten sensitivity will create inflammation throughout the body and cause a whole list of diverse symptoms unrelated to celiac.
Okay. Jonathan, you mentioned that one of the causes of increased food sensitivity could be due to increased histamine in the immune system. Can increasing our DAO help? And how can we test DAO levels?
Right, so the key thing to remember with DAOs, you're really dealing with DAO is an enzyme that helps break down histamine, by the way, and people have used that supplement to help them with histamine reactions. And you can measure your DAO levels with lab tests. But the key thing with DAO is you're really dealing with allergens, with IgE, and not necessarily food sensitivity. You can have very little effect dealing with food sensitivities that are IgG, IgM, or IgA mediated with any kind of DAO supplement. So be aware of that. And a low histamine diet is not going to have much of an impact on people that have multiple food sensitivities because the immune response is beyond just histamine with delayed reactions. If you have a true classic allergy, IgE, and you react to things like dust or pollen or even foods, that's where antihistamines, DAO, what's also really effective is quercetin as a flavonoid. That can really help quench that histamine response. But just DAO pathways and histamine-free diets are really more specific for people that have an exaggerated IgE mast cell response, which is a different immune response than food sensitivities. And you can have both, by the way.
Okay, perfect segue. Magda, what's the difference between IgG, IgM, IgE, IgA?
So what's different is that there's different shaped antibodies, and these different shaped antibodies generate different immune responses. So IgE is your classic allergy. It's an immediate onset response.
IgE like elephant.
IgE like elephant. Thank you. E as in elephant. IgG as in goat. IgM as in maple. IgA as in apple.
Okay.
Those are still bad. Those are delayed, so they take longer to respond. And those are involved with food sensitivity. So IgE is your mast cell, histamine, DAO pathway, and food sensitivity, IgG, IgM, IgA, is your delayed, slow, chronic inflammation pathway that isn't as strongly dependent upon histamine and mast cells.
Okay. Shelby, what is the best way to tighten back up junctions, leaky junctions?
What's the best way to tighten up intestinal junctions?
Yeah.
So this is a question everyone has in a clinical setting at trying to figure out how to help heal patients' leaky gut. By the way, we're working on we just are finalizing the first draft of a new book on leaky gut that goes over all the research and everything on it, but it's pretty complex. So it really depends on what's the mechanism of leaky gut. So you could have intestinal permeability because your diet's bad. You could have intestinal permeability because you have an infection. You could have intestinal permeability because you have hormone deficiencies. You can have intestinal permeability because your microbiome is not diverse. You can have chronic intestinal permeability because you don't have healthy immune tolerance. So at the end of the day, you really have to find what the underlying mechanism is. The simple model you'll find all over the internet and everyone talking about is you have to go on a set of supplements, usually high amounts of L-glutamine, to heal your gut. And if you go on a gluten dairy-free diet and take some glutamine, then that should help heal your gut. And for some people that is true, but for more people it's more complicated. So you have to kind of treat leaky gut as from a whole person approach. What are all the factors that are causing inflammation? Because all the factors that cause inflammation can actually break those tight junctions. So for example, people that are diabetic and have uncontrolled blood sugar and have glucose levels that get oxidized with have high glycated hemoglobin or high HbA1c on blood tests, that glycated hemoglobin will break down their tight junctions. So in that scenario, you can take all the glutamine in the world you want, but unless the blood sugar levels calm down, the oxidation of glucose doesn't calm down by controlling their diabetes, they're not going to be able to resolve their intestinal permeability. So that's the real answer of how these things are. The basic answer you're going to hear all over the internet is take a bunch of nutraceuticals, L-glutamine, probiotics, and get off some inflammatory foods. And that will have some benefits to some degree, but it may not be enough to correct it if there's underlying issues that are causing it.
Okay, Nina, what do you think of microbiome testing like Biome that tells you what foods are best or worst for your system?
I think bio and microbiome have a long way to go. I think there's some big assumptions that they're making, and I don't think they're there yet with their technology and with the models that they're portraying.
Okay. Sean, if one has diligently avoided gluten, soy, casein, and albumin for years, but still tests positive for all four via salivary SIGA, what does that mean?
Well, if you avoid all those food proteins diligently for all those years and you're still showing positive, it's very likely one of two things. You had lab error. The lab that ran your test gave you the wrong results. So it's worth repeating. The other possibility is you're getting trace exposures that you may not know about. And trust me, there are trace amounts of some of these food proteins that may not be on the labels of ingredients. So you could be, for example, eating, I don't know, veggie burgers, and within the veggie burgers, there's trace amounts of these proteins and how they configure it, but it's too small of a dosage to list on the ingredients label, but you're still getting exposure and causing those inflammatory reactions. So if you retest with a lab and you see it's still positive, and you can rule out that it's not lab testing error, you may be getting exposed to food proteins that are not that you're not aware of. And a lot of processed food, especially a lot of processed vegetarian food, has these egg, soy, milk, and wheat proteins in their ingredients that are very subtle. So be aware of that. That's what I've seen in my practice all the time. Okay.
Okay, so the question went away, but I remember it. Priya said, can one follow the AIP diet if they don't eat meat, if they're not a meat eater? Not vegan, but not a meat eater.
Can you follow the autoimmune paleo diet if you're not a meat eater?
If not a meat eater, yeah. Priya.
Not a meat, so vegetarian.
Vegetarian, yes.
Okay.
I'm reading
I know. I'm just trying to process it myself.
Not vegan, vegetarian.
Okay. It's going to be extremely difficult because when you get rid of lectins, if you get rid of lectins and you get off grains, it's going to be very hard to get some complex proteins. So I think you're going to find it, and it's not going to be very effective, so you're going to struggle with it. And food proteins tend to be the least antigenic protein for dealing with what the scenarios are. There's a point, like you see with patients that have autoimmunity and chronic inflammation, they go autoimmune paleo, then they go vegan, and then they go vegetarian, and they go antihistamine, and they have nothing to eat, and their microbiome diversity goes wrong, and they don't know what to do. Like, there's a point you have to settle with something. So I think when you start going in autoimmune paleo and adding things on top of that, you're going to make it extremely difficult to get proper nutrition and proper protein intake in. So I would just say that's my observation.
Okay. Brad, have you ever seen someone not react to gluten and dairy on lab tests?
Yes, there are people that do not react to gluten and dairy. I know it's hard to believe for those of you that are chronic diseases, but people don't. People that have chronic autoimmune diseases, chronic inflammation, chronic inflammatory conditions typically do. But there are people that do not react, and not everyone is sensitive to those food proteins. So that is true.
Okay. So Genevieve, not to be controversial, why are genetic modified foods bad? There are lots of disagreements in scientists about this.
Yeah, it's not to say that all genetic modified foods are bad or good. That's not the issue. I think you don't want to make it black or white. When you genetically modify food, you change the protein structure, and that protein structure change for some groups, for some subgroups, may make it more reactive than others. So that's the concept of immunology. We tend to have some degree of immune sensitization, immune tolerance to environmental proteins our immune system is used to dealing with. So the term they use in immunology is neoantigen, a new antigen. A neoantigen to the species, just like a new virus to the species, tends to potentially cause a higher degree of inflammation in some subgroups, but not everyone. So with people that typically have autoimmune disease, they just realize they can't eat things like soy or corn. They don't need a lab test. They eat it and they fall apart. It's not even, it's not about for the entire human population, but for their uniqueness for themselves. They have a serious problem with it. And I remember when I was out of school and in my 20s, late 20s, and I was working with chronic patients, even from then, just because there's so many of them around. Not because I had the knowledge set to work with them, but they were desperate and needed help, and I was willing to listen and try. These patients would come in with zero doubt they cannot eat GMO foods because they would react to it, and they would bring in their own food because they knew that after the appointment, they need to get lunch. There's no way they could eat out. They just couldn't eat processed foods. When people get some significant degree of inflammation, illness, they can't handle it. So, the most common foods people can't have reactions to tend to be, if you go into an office with an autoimmune disease patient, you start asking them, hey, which foods have you really noticed cause a problem for you? You're going to see soy right up there with corn. Then you're going to see wheat, milk, egg protein, and lectin foods, lectin and nightshades. So it's not about a controversy. It's just that GMOs change the structure of the protein, and new antigens for some groups, especially those that have lost tolerance, can to be a very common trigger.
Okay. So this is from Maggie, but there's quite a few of these questions.
Oh, and by the way, when you take a whole population of people and do a study where you have many people that don't react to GMOs and only have a small person react, when you do actually do the stats, it looks like there's no effect. That's why you have to have subgroup analysis studies done. I'm sorry.
That's okay. Okay, so a lot of people ask this. Do you believe in muscle testing to find food sensitivities? There's a lot of different variations of that question, but that's the basic.
Yeah, I mean, I think in the word science, the word believe is always a bad word. Mm-hmm. The best question is, do you have evidence for? Do you find it clinically useful? Those are the kind of things that are there. But I understand the word believe. It's the term I use myself too. But just to clarify, the appropriate concept is there evidence for and so forth. So I think it's a very controversial topic. I have some experience with biokinesiology myself when I was first in school, and you can definitely see muscle testing changes for various reasons. You could have people think of various thoughts, and muscle testing can change. The tester can have an impact on muscle testing functions. So I think for some people, it's whether some scenarios you're at option of I'm either going to guess one thing or another. If it's just pure guessing with one thing or another, and you've already exhausted labs, or it's not even an option, then maybe that can give you some information to it. So I know there was a paper that was published by Wally Schmidt a few years ago. It was published in International Journal of Neuroscience, where they found some degree of correlation with a competent person that did that. But that's an expert level study participant that's doing it. There's a lot of factors there. So I could say clearly, as a person, from the scientific evidence, they have not established that. But that's not the world of clinical practice, and sometimes there's some anecdotal things that may provide some use. So that's the best I can do to answer it.
Sorry, dog came in. Do you think the GAPS diet is useful for healing leaky gut?
Do I think that GAPS diet is? Yes. I mean, GAPS, again, you can do things like a GAPS diet support leaky gut. Some people are not going to respond because if you don't find the cause or trigger, you're not going to fix leaky gut just by a diet or a nutraceutical. You're going to fix leaky gut by trying to figure out what the triggers are and removing them. If your triggers are just foods you're eliminating and you add in a change in diet and it resolves, that's great. For majority of people, it won't be that simple.
Okay. Can hypoglycemic-like symptoms be tied to food allergies?
Can hypoglycemic-like symptoms be related to food allergies? Well, hypoglycemic symptoms are shaky, lightheadedness, irritability. Of course, it's possible. I don't know. I can't tell you what the physiological mechanism would be. If a patient came to my office telling me that, I'd have to really think about and dissect what they're referring to as hypoglycemia symptoms and kind of work my way backwards. So I'm sure it exists for some people. I just can't tell you just in this Q&A if I know what's causing it.
Okay, Marcia. This is a common question too. How does one go about eating a diverse diet when there are only 3 foods that are tolerable?
Right. So this is something we talk about in our immune tolerance program, in the 3D immune tolerance program. When you actually lose immune tolerance and you do food sensitivity and test for every food, you can't just eat those 3 foods. You're going to actually make yourself worse. You're going to lose your microbiome diversity. So as a clinical example, let's say someone comes in, they have multiple food sensitivities on a panel. We test, let's say, 200 foods, and more than 50% of them have a reaction. If we just have them eat the foods they don't react to, and and follow up testing with them in about 3 to 4 months, all the new foods they're eating, they're going to have sensitivities to it, and then they'd have more and more food sensitivities. And if you only let rant into 3 foods, it's probably that's you may have gone through that, because that's what happens when you do that. At that point, what you have to do is you have to look at foods when you lose your immune tolerance and you're reacting to everything, every food protein. What you may need to do is really look at strategies to improve your dendritic cells, things like leaky gut, improve your IgA levels, and also you have to eat foods. You have to eat the foods you don't react to. So what I'll do in my practice if I see persons react to everything is I go, well, you're going to have to eat a diverse vegetable diet. So we encourage them to eat all of their vegetables in a diverse fashion, eating a lot of diverse vegetables. I avoid foods that have nightshades or lectin, vegetables that are lactose and nightshades, because they're so inflammatory for these people that are chronically inflamed and have lost their tolerance. And if they don't have any symptoms, for example, they may show up with carrots or Brussels sprouts, but when they eat it, they don't notice anything. It's just showing up in the lab test. At that point, it's important just for them to eat it and as many other vegetables as they can that are not nightshades or lectins to really improve their microbiome diversity, to then change their immune tolerance so they can start working their way out of being so sensitive to foods. This is the whole concept of immune tolerance. And this is why I created a program for it, because everyone is just fixated on leaky gut, but it's not that simple. And if you just focus on leaky gut and constantly removing foods, you can't turn this around. So the concepts of immune tolerance that I put together in the program is the 3D: diversify, distinguish, and downregulate. Again, you can go to Dr. K News. We have articles and online program about that. Okay, any?
Here's another one.
One last question.
Okay, from Rebecca. Other than gluten and the cerebellum, are there specific food antibodies that have cross-reactions with specific areas of the brain?
Yes.
Okay, done. Just kidding.
There are food proteins that have reactions with, there's a paper that we published in Journal of Diabetes Research. We looked at GAT65. GAT65 is one of the main autoantibodies in the basic ganglia in the cerebellum. You can go to PubMed, National Library of Medicine. If you type in my name and look up paper on polyclonal monoclonal antibodies to the thyroid axis, we list those food proteins. And that's a really common autoimmune target site for chronic anxiety and for chronic what I call basic ganglionic disorders. So you may want to check that out. But wheat germ agglutinin is strongly cross-reactive to different proteins in the brain. Gluten is significantly cross-reactive to the cerebellum, like you talked about. And then we published a paper in the journal Nutrients back in 2014, I believe, where we looked at milk protein being significantly reactive to myelin. I think those are the main factors that we know about so far. Anyways, I hope you found this useful, and I hope you can enjoy these as we do them. And thank you for your time, and thank you for joining us today.